Greetings, dear colleagues. I'm going to talk about the role of immunology in physiology. Some years ago, medicine was made of chemical compounds. Medicines treat broad diseases, and we used radiation and chemotherapy to treat cancer. But now medicine is made from living cells. Medicine starts to specific patient based on genetic makeup. Also, method of treatment is immunotherapy that harnesses body's own immune system to fight disease. Also, we use CAR T-cell therapy. It's a way to get immune cells, called T-cells, to fight cancer by changing them in the lab so they can find and destroy cancer cells. Next method of treatment is CRISPR. It's a genetic engineering technique in molecular biology by which the genomes of living organisms may be modified. Currently, one of the main global trends in the development of pharmaceuticals is personalized medicine. It includes uses diagnostic tools to identify specific biological markers that determine the optional therapy methods for a particular patient. For example, 42% of new drugs under development will potentially become personalized drugs. 73% of cancer drugs under development will personally become personalized. Investments in the development of personalized drugs are expected to increase by 33% in the next 5 years. The number of personalized drugs being developed is expected to increase by 69% in the next 5 years. Key measures to achieve the end of tuberculosis strategy goals by 2030 and 2035 includes prevention of new causes of Mycobacterium tuberculosis infection and their progression to tuberculosis, and also identification and effective treatment of people with latent tuberculosis infection. Latent tuberculosis infection is a state of persistent immune response to Mycobacterium tuberculosis antigens and that have previously entered the body, the absence of clinical manifestation of the active form of tuberculosis. In this slide you can see two types of tests which is used for early detection of tuberculosis: skin and laboratory tests. The basis of immunological tests for early detection of tuberculosis infection. The antigen were taken from the RD1 region of the genome Mycobacterium tuberculosis. If the RD1 site is absent in all BCG strains, the test won't lead to false positive results in BCG vaccinated patients. If the RD1 site is absent in non-tuberculous Mycobacteria, the test won't lead to a false positive result in mycobacteriosis. What's the difference between dye skin test and Mantoux reaction? The basis of the skin test is HER2-specific proteins, which are only present in infection Mycobacterium tuberculosis. The basis of the Mantoux reaction is tuberculin. It's a mixture of more than 200 antigens. In accuracy of diagnosis of tuberculosis infection, the specificity of the test is 98.5%, but Mantoux about 30%. The sensitivity of the test is 98.3%, but Mantoux reaction is 98%. The second, there are no false positive results in BCG vaccinated individuals. The Mantoux reaction can have positive results for BCG vaccine. It's necessary to do additional examination if there is a positive result. And the last feature of the skin test and Mantoux reaction is high pharmacoeconomical efficiency of tuberculosis infection screening. There are two laboratory tests which we use to determine tuberculosis infection. The principle of the method is that open contact of specific Mycobacterium tuberculosis proteins with T lymphocytes of the infected patient's blood, and gamma interferon is released. The test measure includes the amount of gamma interferon isolated, its ELISA method, and second, the number of cells releasing gamma interferon, its ELISpot method. According to the presented publication, the T-spot test has a higher specificity compared to the ELISA test, including in patients with immunodeficiency, and also it provides a minimal percentage of uncertain results. What's the difference between ELISpot and ELISA? The T-spot test is more highly sensitive than QFT. Sensitivity of the T-spot test doesn't differ significantly with age, while QFT sensitivity decreases after 30 years. T-spot has two additional laboratory steps compared to QFT. White blood cells are isolated and washed to remove unnecessary substance. Standardization of samples by the number of lymphocytes includes the presence of false results, especially in patients with immunosuppression. Against the background of using T-spot test, there are a low percentage of uncertain results. Some medication, for example antidepressants, may reduce the release of interferon gamma by T lymphocytes. When taking the T-spot test before the start of antigen simulation, T cells are washed in impurities of any drugs are removed. Standardization of sums by the number of lymphocytes leads to the fact that the result isn't affected by the immune status of a person. Sensitivity of the T-spot test in HIV-infected person exceeds all available tests. Modern tests for tuberculosis immunodiagnostics include the skin test and T-spot test. And the skin test is a skin test with tuberculosis recombinant antigen. It responds in detection of papule at the injection site in the presence of infectious Mycobacterium tuberculosis in the body. The specificity of the skin test is 98.5%. The skin test sensitivity is about 98.3%. It's used in screening for tuberculosis in children and adolescents. The T-spot is determination of the number of activated T lymphocytes releasing gamma interferon. Spot indicates one activated T lymphocyte cells. The specificity of T-spot is about 100%. A sensitivity of T-spot is about 98.8%. The reason doesn't depend on the level of immunosuppression. T-spot localized in Russia in 2019. To make a diagnosis of primary immunodeficiency, we can use for the study whole blood or dry human blood strains. The principle of the method is quantitative determination of the content of DNA cubes of and crack in nucleic acid samples by polymerase chain reaction with hybridization fluorescence detection of reaction products in real time (PCR-EV). A set of reagents is used as an auxiliary tool for the diagnostics of pathological conditions caused by a lack of T and B cells in groups of the child population without gradation by demographic or population characteristics. The presence of immunodeficiency is an absolute contraindication for the tuberculosis vaccination. However, on the 3rd to 7th day, when BCG/BCG-M vaccination is carried out, it's clinically difficult to establish the presence of congenital immunodeficiency since there are no characteristics manifestation of the disease yet. They use a big test before using live vaccines, in particular BCG, can prevent serious complications of vaccination. The new technologies helps us to make laboratory research methods or instrumental research methods that we need. For example, chemical or method of recombinant DNA or blood component test or immunological preparations. New technologies helps us to make a diagnosis with all types of laboratory or instrumental research methods. For example, chemical tests or method of recombinant DNA, biomedical cell products, and other. The active agents which are produced in our body are antibodies, cytokines, interferons, and interleukins. It produced when we have an infection and after vaccination. Vaccines are divided into 4 types. The first type is inactive or live attenuated vaccine. This type of vaccine uses a weakened form of the gen that causes a disease. Because these vaccines are so similar to the natural infection, they help prevent. They create a strong, long-lasting immune response. The next type of vaccines is VLP vaccines. It stands for virus-like particles. Virus-like particles are molecules that mimic viruses but are not infections. They are a very effective way of creating vaccines against disease such as human papillomavirus, hepatitis B, malaria, and more. The next type is viral vector vaccine. It's a vaccine that uses a viral vector to deliver genetic material coding for a discrete antigen into the reception host cells. And the last type is vaccines based on proteins, DNA, and RNA. They consist some proteins or RNA or DNA of pathogen bacteria. There are some types of drugs which was repurposed for treatment of coronavirus infection. For example, baricitinib we use to treatment of rheumatoid arthritis. Favipiravir we used for treatment of other viral infections. Or ribavirin, for example, we use to treatment hepatitis C or some viral hemorrhagic fevers. All of these drugs affect on the mechanisms of human immune system. So it's really important to choose a right method of treatment or right method of therapy or right medicine. Dear colleagues, thanks for your attention. Goodbye.